So I think the slides are visible and I am also properly audible. So good evening everyone. Welcome to the week 9 section or extra section for the topic cell culture technology that is being conducted by NPTEL and for my PMRF TAship that is being conducted by IIT Madras. So we will just move on to the first question itself. Moving on to the first question, that is question number 1. Muscular dystrophy causes gain in muscle mass, progressive muscle weakness, both A and B or none of the above. Which might be the answer? You can either type in the chat box or you can just unmute yourself and answer the question.
Option B.
Option B. Okay, any other option? Okay, so basically you are right. Muscular dystrophy causes progressive muscle weakness. So basically it is characterized by progressive muscle weakness as we have discussed here. So it results in some genetic mutations that are happening within the body or the skeletal muscles that impairs the production of proteins essential for muscle health, which in turn leads to muscle degeneration, and these proteins might be involved in maintaining the muscle integrity or structural and everything like that. So basically if you look into a deep dive effect, basically you don't need to know the advanced section, but I'm just pointing some of the points out. So if you look into a muscle dystrophy and all, basically it's basically genetic in nature. So mutations happen in genes that are basically associated with S chromosomes and all. So basically this is characterized by muscular dystrophy is characterized by a group of different type of muscle degenerative disorders. Examples if I give, Becker's is there, Duchenne muscular dystrophy is there, and several others are also there. So basically they are related to mutations that are happening in genes that are present in S chromosomes basically. So these are most probably prevalent in case of prevalent in case of like males, because if you look into males we have SY chromosome, while in case of females there is a 2X chromosomes, right? So basically if mutations happen within the genes of S chromosomes, then in case of females there is another copy of S chromosomes that you might have received from other parents that might be able to sustain that particular loss there. But in case of in case of males, what happens is there is only one S chromosomes there, right? So if any mutations do happen in the genes associated with S chromosome, that will be more progressive in case of males as well in this case. So basically that's just an additional point. So in this case, muscular dystrophy causes progressive muscle weakness because of genetic mutations that are happening in genes that code for certain proteins that are involved in muscle development and all, especially in case of skeletal muscles. So does anyone have any doubt, or is this clear? Okay, so I think this is quite clear, right? So I will just move on to the next question. Coming on to question number 2. MEMS stand for hello. Hello, am I audible? Yeah.
It's audible.
Okay. So coming on to question number 2, MEMS stand for microelectrically modified stain, muscular electromodular system, muscarinin epinephrine marking stain, or microelectromechanical system. Which might be the answer?
Option 4.
Yeah, I'm getting option number 4. So basically you are right, it stands for microelectromechanical system. So basically from the name itself, it means that from the electromechanical system, it means that it involves some components which are electrical in nature, electronic components, and others which are mechanical in nature. And microelectromechanical system means that it is synthesized or it is made at a micrometer scale, right? So at that microscopic scale it is actually made. So basically if you look into say this MEM device, it consists of sensors, actuators, or other microelectronics that are needed for whatever is the application that particular system or any other instrument does, right? So it is basically miniaturized into a microscopic scale. It is often small in size because of that, and the mechanical as well as the electronic parts also has to be made or microfabricated according to that particular range. And integration means that these components, that is electronic as well as mechanical systems, are integrated within that particular mechanical range in that case. So or it is integrated together to perform that particular application whatever it is supposed to be carried out. So examples include like micro cantilever system that are required to study the muscle movements and all, whether the muscle is properly functioning or if there is any other studies that you need to carry out and all. So I hope this answer is clear. So anyone has any doubt? Okay, so I think the answer is quite clear. So I will just move on to the next question. Coming on to question number 3. A measurement of generated force can be recorded for the following cell type to assess their physiology. First one is neurons, cardiomyocyte, beta islets, or astrocytes, which may be the answer.
Option 2.
Option 2, cardiomyocyte. Any other option? Okay, so basically you're right. Cardiomyocyte, or also called as heart, right? Heart cells, right? Cardiomyocytes means heart cell or cardiac cell. So basically, if you look into when a neuron or anything that a motor, suppose that a motor neuron is associated with the skeletal muscle, so you have received a sensory signal or you have received from a signal that from a brain. Basically, based on that signal, the motor neuron is activated. The motor neuron in turn sends a particular signal to the neuromuscular junction that is associated with muscle fibers, right? Skeletal muscle fibers, because they are involved in movements, right? So when it sends a signal, the muscles get activated. So you know that there are several muscle proteins that are associated, right? Myosin is associated, actin is there, and there is also a protein called titin here that actually integrates this myosin with the actin filaments there. So basically, upon receiving this signal, the muscle starts to contract. So basically, there is an electrical signal that is happening with the neuron. Then it is transferred to a chemical signal at the neuromuscular junction. Then it is transported into a muscle fibers that is activated. So it results in this contraction, or the sarcomere length actually gets reduced in this contraction there, right? So basically, when this happens, because of the electrical force that is generated from the neuron, which is converted into a mechanical force, the muscle movement, right? So upon the muscle movement, so when the muscle start to contract, it generates a force there. That force is the particular reason that is produced because of the sliding of the myosin over the actin, right? So this particular force can be recorded in to assess a particular cell type, to assess their physiology. Basically, leave the cardiomyocyte. Cardiomyocyte is actually like a muscle there, right? Cardiac muscle there. Smooth muscle also generates these kind of forces. Skeletal muscle also generates these kind of forces. And if there is any genetic disorders that are specifically associated with that particular muscle cell, then that particular force that is being generated there also differs. So this kind of force actually gives the value of the force that is generated gives a assumption of what kind of a cell type or a muscle cell type that is being that you are basically studying there. It gives you the identity or a characteristic of the muscle fiber that you are basically studying there. So in this case, neuron is involved in electrical transfer, electrochemical activity, right? So it doesn't generate a force. Cardiomyocyte actually generates a force because it's a muscle system. Beta ILS is actually not associated with this. And astrocytes also, it is found in like central nervous system, right? Astroglia cells are also found in central nervous system, which are involved in like defense activity, immune response and all. So basically the answer to this question is cardiomyocyte, and the measurement of generated force can be recorded for which cell type to assess their physiology? Cardiomyocyte. Other muscle types can also be used to generate force from other muscle types can also be used to study their physiology basically, not only cardiomyocyte. So anyone has any doubt regarding this? So I think this is clear. So I will move on to the next question. Coming on to question number 4. For a micro cantilever based system, Stoney's equation correlates the following two parameters with each other: deflection with force, deflection with cell type, fluorescence with force, fluorescence with cell type. Which might be the answer?
Option 1.
Option 1. Any other option? Yeah. So basically the answer is you are right, deflection with force. So basically this is a cantilever system. It is at a microscopic scale. Leave the other designation that is here, pump, probe, everything, leave that, the mathematical equation, leave that. So basically it's like a swimming board, right? So basically if you have watched the diving program or anything like that, basically a swimmer basically or diver stands at the end of this cantilever, and then they jump up and down to get the thrust. Then afterwards they are diving into that particular swimming pool, right? So basically this particular cantilever is also called as micro cantilever because it is at a micro, it is made at a micro level scale or micrometer scale. So basically if you look into a cantilever system, you can see that it actually oscillates, right? So basically, as I said, when a swimmer jumps up and down, this cantilever also oscillate along with that, and that's what gives the thrust to a swimmer basically. And also based on this flexibility, and because this cantilever is basically flexible and it is only in contact or fixed at a particular point, right? So other points are free. So whenever you give a force here, it oscillates up and down for a particular minute, then it comes, then it stops afterwards because of the friction or the air resistance there, right? Basically if you are providing it at a micrometer scale, basically you can you can be used to study muscular movements and all. So if you are putting a skeletal muscle, suppose that I am taking a skeletal muscle here, cardiac muscle here, so it been fixed from this one point to the other point. So if the cardiac muscle is active, then what it does is that it contracts and relaxes, right? So when it contracts as well as relax on over that particular period of time, this cantilever also starts to oscillate, and this oscillation takes place at a particular angle. So because if it is active, right, it continues to oscillate within a repeated stage, and it is particularly constant in that oscillation will be constant in nature. So that particular force generated will allow the cantilever to oscillate up and down. And when you are comparing that oscillation from this fixed point, if you are comparing an angle from this particular fixed point to this fixed point, then if you particularly get to know that particular angle, then you can get to know how much force is being generated there. This particular equation is also called as Stoney's equation. So from that particular theta that you are getting, if you are putting that into a particular equation, then you are getting a particular force that is being generated there from that particular movement that is caused by those skeletal muscles or cardiac muscle, whatever you are typing. So basically that deflection or that angle is called as deflection there, because that deflection, that angle measurement gives you a assumption or a measure of that deflection that is happening there, and from that deflection that is generated there, you can actually calculate the force there. And from the earlier question that I said, from that force value that you are getting, you can actually get an idea of what type of a muscle it would be, whether that muscle is properly functioning or not. So that's is basic functioning of a micro cantilever based system. And also, can you based on the fluorescence also you have this particular option there. Can you give me an idea of what this designate, which type of application would this contribute to, fluorescence with cell type? What kind of instrument might be using that? Fluorescence with cell type.
Tagging fluorescent molecules with
Yeah.
uh,
Yeah.
bind to the cell type receptors.
Yeah. Basically, flow cytometry or FACS, right? Basically, we are tagging that with a particular antibody there, and then we are based on the fluorescence that you are getting, you can sort it out into different vials, right? So that's one common example. Or basically immunofluorescence can also be used, because if you are using an antibody which is fluorophore, that can bind to specific receptors that are present on the cell surface, right? And then you are observing it to it, then you can distinguish between different cell type also. So that's also one example. Okay, good. So does anyone has any doubt regarding this? Okay, so I think this is quite clear. So I'll just move on to the next question. Coming on to question number 5, which of the following antibodies can confirm the formation of skeletal muscle myotubes? Option number 1, connexin, MAP2, myosin heavy chain isoform, or chat, which might be the answer.
Option 3.
Option 3. Yeah. So the answer to this question is option 3, myosin heavy chain isoforms, right? So basically myosin is a component of like muscle fibers, right? So if you look into this particular figure, you can see that the dark band is contributed by myosin there, while the light band is contributed by actin there. So once actin and when signal is received, the myosin binds to these actin filaments there, and then it pulls the actin towards this particular central line, right, M line. So that results in contraction. While in this case, when the myosin is not bound to the actin filament, that is particularly called as a relaxed phase of a muscle fiber, right? So that is also, this particular theory is also called as the sliding filament theory. So myosin as well as these other proteins, actin, and this particular protein is called as titin there. So if you are antibody that can bind to either actin, either myosin, or what you say, titin there, then you can get an idea of whether it's a skeletal muscle myotube or not. So if you want to specifically go into other cell type, then you might have to use other antibodies that could bind to specific receptors. That's a different thing. So myosin is the answer there. And if you look into connexin, basically it's associated with gap junction and cell communication there. So it's not associated with skeletal muscle. It can be associated with the other type of like what you say, muscle fibers or any other particular protein, or it can be universal in nature. MAP2 or microtubule associated protein 2 is actually associated with the neurons, right, particularly in dendrites. So it can be used to, if an antibody that can detect MAP2, it can be used as a particular detection method to identify neural cells and not muscle cells. And that also called as choline acetyl transferase. And so acetyl, it is used in like in acetylcholine neurotransmission and all, transmitter activity, generation of acetylcholine and all. This particular type of this particular enzyme is used. So it can actually, if any particular antibody is used against this particular enzyme, it can be used to study the what is it, cells that can interact with acetylcholine and all, or the neural fibers that are basically cholinergic in nature. So, and also if you look into microtubule acetate protein 2, this we have discussed earlier also, but I am just pointing out so that you can just recall out. So it is used for labeling neuronal cells due to specific localization and functional property within the cytoskeleton of neurons, and it is associated with the cytoskeletal stabilization, maintaining the structural integrity of neurons there. Microtubule dynamics, because in order to form microtubule neurons and all, this is specifically involved. Signal transduction, and also in case of disease pathology like neurodegenerative disease like Alzheimer's or Parkinson's, there will be change in this particular MAP2 protein levels and all. So yeah, so I think the answer to this question is clear. Which of the following antibodies can confirm the formation of skeletal muscle to myotubes, which involves myosin heavy chain isoforms? Do you have any doubt regarding this, or should I explain it once more? Okay, so I think this is quite clear, so I will just move on to the next question. Coming on to question number 6. To chemically modify the substrates in order for the cells to not grow, one should use cytophobic material, water, alcohol, which might be the answer.
Option 2.
Option 2. Yeah, I'm getting option number 2. So the answer to this question is cytophobic material. Basically, cytophilic materials involve majority of the cells like media that we are using, the extracellular matrix that we are providing, or any other component or any other material that probably promotes the growth of this particular cell, right? So cytophilic means cell-loving material, right? So cytophobic means it hates the cells there basically. So it prevents the growth of cells there. So basically some of the cytophobic material are designed to repel cells, preventing their attachment and proliferation on the surface. So if you look into any kind of adherent cells, right, it requires a extracellular matrix for it to attach and then proliferate, right? So if somehow preventing the attachment there, then those kind of cells won't grow. So examples include polyhema or 2-hydroxyethyl methacrylate, and this restricts the cell adhesion, and which in turn prevents the growth there. So in case of adherent cells, these are probably used there, and it is also non-toxic in nature. Basically, if we are using something like a microfabrication, like if we are using a micro cantilever system which promotes the cell growth at the surface, and it also consists of a small depth there, a dish like structure there, where you are providing a media, but you don't want the growth of media at that particular surface. You only want the growth of the cells at that particular surface, or that particular depth there, or that particular well there, but you only want the growth of that particular cells or the cantilever there. So in that case, the dish or the dish that is present below the cantilever there is coated with this particular kind of cytophobic material there. So only the cells will grow at the cantilever, not on the particular dish, because of the presence of this polyhema component there. So if you are coating with cell, if you are affecting the cells there, it might affect the whole system properly there, right? So in that case, you need a substance that does not harm the cells, which is not toxic to the cell, but at the particular surface where it is coated, it will not kill the cells there. So basically, polyhema is one, the 2-hydroxyethyl methacrylate is one such component that prevents cell adhesion, and it repels cell there. So basically, it inhibits their ability to attach and grow. So that's one such example, that is polyhema there, which is a cytophobic material that prevents the attachment of cell surface to the extracellular matrix or any other particular surface there, and then in turn prevents it to grow, in turn affects the growth of the cell, or it reduces the growth of cells there. So I think this type of material is used in micro cantilever system that we have earlier discussed before, right? So I think this is clear. Anyone has any doubt regarding this? Okay, so I think the answer is quite clear. So I will just move on to the next question then. Coming on to question number 7. Which of the following is used extensively to make this microelectromechanical system or MEMS device? Polydimethylsiloxane, N1,3-trimethoxysilyl propyl diethyl triamine, diethylene triamine, trimethoxy 333 trifluoropropylsilane, or dimethyl sulfoxide. Which might be the answer?
Option 1.
Yeah. So the answer to this question is option number 1, polydimethylsiloxane. So basically, this is the structure of the the polydimethylsiloxane. Basically, it's a polymer, right? From the name itself, you get an idea that it's basically a polymer there. And basically, polymers are used to make this kind of MEMS devices, but not all polymers. So it should be compatible in nature. It should be flexible, biocompatible, ease of processing. This all applications it should carry out basically. And polydimethylsiloxane is one such polymer that have this kind of advantageous properties, and hence are used to extensively make this kind of MEMS device or micro electromechanical systems. And you can see that there are methyl groups that are attached to this silicon group there. There's an oxygen group also there, and this particular is the repeating unit of that particular polymer there. and at the ends you can see that there is a silicon that is attached to 3 methyl group there. At the other end also you can see the similar trend there. There is a silicon group that is attached to 3 methyl group there and is conjugated to this particular repeating unit by an oxygen group there. And you can see the representation number there. It can be as much as length as it want because it is a polymer basically. So answer to this question is polydimethyl siloxane. Which of the following is used to extensively make MEMS devices? Polydimethyl siloxane, because of this particular advantage that is usually has. So I think this is clear. So do you have any doubt? Okay, so I think I will just move on to the next question now. Coming on to question number 8.
Sir,
Yeah.
sir, others material were used.
Could you please repeat the question? I wasn't able to hear it properly.
Sir, the other material mentioned in the options, where are they used?
Okay, basically I haven't checked that properly in this question. Dimethyl sulfoxide, I know basically is a liquid solution there. It's basically used to, if you are doing any protein work or if you want to solubilize any drug, basically it's used as a universal solvent there. So as much as I know, it's not a polymer there. And also, yeah, basically it's used to dissolve drug and it's basically a universal solvent there. The other such components basically I have to like check out. Basically at this point I don't know the proper answer to that question.
Okay, sir.
Okay. So any other doubt there? Okay, so I think it's clear. So I will just move on to the next question. Coming on to question number 8. For a cantilever to bend upon muscle movement, which of these properties of the cantilever material and device should be carefully decided? One, color, insulation, elastic modulus, or electrical impedance. Which might be the answer?
Option 3.
Option 3, right? So elastic modulus basically. So basically it's a unit of measurement whether the component or the substance or the surface that you have just made is how much it can be deformed based on the elastic nature that it can be performed, right? And if you look into cantilever system, right, basically I will just move on to the previous slide there. Okay. So look at this image there. Basically, if you putting a skeletal muscle or any type of single muscle cell at the top, right, it can contract basically. So basically, a cantilever system should have the capability of having a proper flexibility to oscillate in accordance with the skeletal muscles that is getting contracted and as well as relaxed there. So basically, if it doesn't possess this characteristic, then it will create a problem so that we cannot basically calculate the deflection there. If we are not properly calculating deflection there, we cannot actually get an idea of how much force that is being generated there, right? So this particular cantilever system should be flexible in nature. So it depends on this particular what we have discussed there, right? So elastic modulus there. Elastic modulus is one such equation which is a ratio of, I think, a stress to strain. I actually don't recall the proper meaning there. So whenever a stress is applied, it actually has the ability to deform itself. Not only deform, it has to reform itself and then carry out that particular oscillation a number of times. And from that particular oscillation, an angle is generated there. From that angle, you can basically get an idea of the deformation, which in turn gives you an idea of how much force that is being generated by the muscle movement there. So basically the nature of the material that is being made there, when that is being used to make the the cantilever there, depends on there, and also the thickness of the cantilever. So if you are able to make a fine thickness of the cantilever, then it's good because it can actually fluctuate on the basis of the force generated there, right? But it shouldn't be that thick as well as it shouldn't be that thin, because it should not break as well based on the force that is being generated as well. It should also oscillate at a particular optimum range based on what we could make a particular measurement from there, right? So there should be an optimum range there. And so elastic modulus is the unit of measurement of that particular object or substance, in this case that cantilever, resistance to being deformed elastically, that is non permanently. It should reform back, as I said before, when a stress is applied to it. The stress is because of the movement that is being generated by the contraction as well as the relaxation of the muscles that are present in the cantilever there. So yeah, that's the basic answer to this question. For a cantilever to bend upon muscle movement, which of the properties of the cantilever material and device should be carefully decided? So the answer to this question is elastic modulus. So I think this particular question is clear. So anyone has any doubt regarding this? Okay, so I think the answer is quite clear. So I will just move on to the next question. Coming on to question number 9. Adding this basic fibroblast growth factor or BFGF to skeletal muscle culture, one can prevent or reduce cell doubling, cell fusion, both cell doubling and fusion, none of the above. Which might be the answer? Okay, I am getting option number 2, cell fusion. Any other option? Okay, so your answer is right. Basically, cell fusion. Basically, if you look into basic fibroblast growth factor, right? So there in coordination with the muscle fibers, there is also called as something called as a fibroblast cells there, right? So if you look into a lecture videos, you would know that whenever you are growing a muscle fiber, you also need to have an appropriate amount of fibroblast cells there, right? It should be in optimum range. It should not exceed a particular value, so that because the proliferation capability of this fibroblast is much more higher as compared to that of a muscle cells there, or this myoblast cells there. So the range of fibroblasts that you are introducing into a culture tissue or a dish should be optimum there, or much less than the muscle tooth there. So whenever this fibroblast grows, right, it starts to release out certain growth factors or any other components that results the muscle fibers to proliferate. So if you are providing just a skeletal muscle, it won't grow. You need to provide the appropriate amount of fibroblast along with it, so that it can secrete some growth factors, which are called as BFGF, or in this case basic fibroblast growth factor, which are involved in this particular division of or proliferation of these satellite cells there. So basically, if you look into a muscle fiber, there are certain cells called as satellite cells. These are basically like stem cells that have capability to generate into a muscle fiber there. So in this case, you can see that there are specific steps that it goes through, right? First, it goes through a formation of a myoblast there. This myoblast actually arranges this pattern, then arranges itself into a pattern to form what that fuses together to form what is called as a myotube. This myotube then arranges itself to form a muscle fiber there. So if you are providing a fibroblast there, then it generates these growth factors there that helps in cell doubling or cell proliferation there. It doesn't promote cell fusion there. So if you are removing these growth factors or the fibroblast, and then you are providing a new media there, then the already generated myoblast patterns itself to form a myotube there, which is a functional unit of a muscle fiber there. So if you are then again providing the fibroblast growth factor there, then chances are that proliferation starts to happen with the remaining satellite cells there, and this particular fusion won't take place there. So it just remain within that particular myoblast stage there without forming into the other downstream pathways, downstream stages of a muscle fiber there. So basically, it inhibits the different. It has been shown to promote a proliferation of myoblast in vitro, meaning it encourages cells to divide and increase in number. But it inhibits the differentiation of myoblast into these myotubes, which is a critical step that characterizes this particular option there, that is the cell fusion. By repressing this differentiation, this particular growth factor effectively reduces the fusion of myoblast into muscle fiber tube, or more probably what you call as a myotube here. So I suppose the answer to this question is clear. Adding basic fibroblast growth factor to the skeletal muscle fiber, one can prevent or reduce cell fusion and promote cell division there. So I think this is particularly clear. So does anyone have any doubt regarding this?
Sir, is the myotubes and myofibrils are similar?
Myotubes and myofibrils, I think myotubes combine to form a myofibril. This myofibril in turn intertangle with each other to form a myofiber, muscle fiber there. So yeah, that might be the answer.
Okay, thank you, sir.
Okay. Any other doubt? Okay, so I will just move on to the next question then. Question number 10. The current view is that every individual skeletal muscle myoblast can, option number 1, proliferate and fuse simultaneously, cannot proliferate, cannot fuse, either proliferate or fuse at a particular point in time. This may be the answer.
Option 4.
Yeah, option 4. Any other option? Yeah, so basically you are right. The answer to this question is either proliferate or fuse at a particular point in time, right? So myoblast has a tendency to proliferate and fuse, increase in number, and fuse combine to form myotubes, but not simultaneously. So first you need to know this step. Basically here I am just repeating it. Satellite cells actually divide and then it forms this myoblast there. So myoblast then combine or fuses, it loses some of this patterning there or cell boundary there. Because of the loss of the cell boundary, these combine together to form in a particular pattern there, which then attains the shape of this myotube there. And it is basically each myotube, if you look into this, is basically multinucleated in nature. And this myotube in turn combines to form a muscle fiber there. So yeah, that's the basic step there. So it then have to first proliferate. The proliferation takes at a particular stage in their life. Then it can actually fuse, and that can also take place in a particular stage in their life. Those two doesn't seem to have happening simultaneously. And as I have discussed from this particular question there, because of the presence of these growth factors or the absence of those growth factors, proliferation can happen or fusion can happen. If you take this a particular example, this particular growth factor, if this is present there, then proliferation of the satellite cells as well as myoblasts take place, and without any cell fusion. But if this growth factor to have been removed there, then this myoblast actually combine together in a particular fashion to form what is called as a myotube there, and then in turn form the functional unit of this muscle fiber there. So that's the basic answer there. The current view is that every individual skeletal muscle myoblast can either proliferate or fuse at a particular point in their time and does not happen simultaneously. And I think that particular answer is clear. So does anyone have any doubt? Okay, so I think this is quite clear. So I will just move on to the next question. So this is basically a question related to action potential and all. Question number 11. During depolarization, membrane potential shift from negative to positive, positive to negative remains unchanged, or none of the above, which may be the answer.
Option 1.
Negative to positive.
Yeah. So basically you are right. Option 1. The negative to positive. So basically this is an action potential, right? So whenever a muscle fiber or any neurons receive any signal from each other, then there is an influx or outflux of particular ions that results in what is called as an action potential, right? So in the resting, this is the resting membrane potential there. We can see that it has a particular range of about -90 in there. So when it is getting activated, what happens is that in the resting stage you can see that the membrane potential at the outside is basically positive and the inner is basically negative in nature. So this actually keeps the transfer of signal along the axons also, right? So whenever an electrical signal is jumping from one position of an axon to the other, then it should undergo a simultaneous stage of action potential, which involves depolarization, repolarization, resting membrane potential, and whatever that is necessary there. So basically whenever in the resting signal you can see that all the membrane channels are closed there, sodium ions are most probably present on the outside and potassium ions are most probably present on the inside, and due to a charge difference or the number of charges that are present on the outside there, you can basically create a potential difference there. Because of the large number of positive ions on the outside, the membrane potential is usually positive there. And whenever a signal is received there, you can see that it undergoes a stage called as a depolarization there. So in this case, what happens is that the sodium voltage gated sodium channels that are present there actually opens up. So whenever it opens up, then sodium, the potassium channels are actually closed there, but the sodium channels are open there. So the sodium ions move to the inside of the cell, thereby making a positive potential difference there. So you can see that that's why the answer is there. In case of depolarization, what happens is that the membrane potential shift from whatever you call as a negative potential gradient that was present in the resting membrane potential to a positive potential gradient there. So that's what's happening in depolarization there. Then afterwards, once the signal has reached this threshold, it starts to die down. So this particular stage is called as a repolarization there. So in this repolarization there, you can see that the channels that are earlier open there were sodium channels there, right? So the sodium channel closes up and then the potassium channel opens up in this case. So this happens simultaneously. Sodium potassium channel opens up when the sodium channels actually was open in the depolarization stage. When the repolarization started, this simultaneous change happens, and in turn the sodium channel which was present there, potassium channel which was present there actually opens up. So the potassium ions that are present inside the cell actually moves out of the cells there. So then there is a shift within the potential membrane potential there, which was positive on the inside there, then it was negative on the outside there. Then there is a shift there. In case of repolarization, what happens is that the positive potential that was present there actually dips down to the resting membrane potential there. So this particular stage is actually resulting in a shift of the membrane potential from positive to negative there, because of the simultaneous opening of potassium channel in the repolarization change when the sodium ions are actually getting open there. So then it moves on to a resting membrane potential there, where the membrane potential that was present in the initial stage were getting actually restored there. So both the channels then get closed there, and then it maintains a resting membrane potential there in this case. So in this case, hyperpolarization is also a stage there when the voltage gated potassium channels then actually remains open for a long time, so more amount of potassium moves out, so the membrane potential actually decreases above below this particular limit, negative value, right? So this particular stage is also called as a hyperpolarization stage there. So in this case, the answer to this question is during depolarization, membrane potential shift from negative to positive. So I hope this answer is clear. So does anyone have any doubt regarding this? This is some basic biology,
Sir,
right? So yeah.
sir, in the figure at the right top,
Yeah.
there they mention that K K channel is also open, but K channel is open towards the inside of the membrane, na?
Yeah, membrane is inside, but the potassium ions are majorly present on the inside, right? So in this case, what happens is that the potassium that is present on the inside moves out of the cell, thereby restoring that potential that is negative resting membrane potential there. While in case you can see that in case of sodium gated channels, there are two gates there, right? So there should be simultaneous opening of both cases so that the sodium ions can move in. But in case of potassium channel, only one gate is there basically. So basically the gate has to be open so that that transition can happen, that movement of or influx or outflux or whatever it is, so the ions can take place basically. Yeah, so I think that's the answer.
Okay, sir.
Okay. So does anyone have any doubt regarding this? Any other doubt? Okay, so we will also discuss the next question. It's also similar. Coming on to question number 12. During repolarization, membrane potential shift from negative to positive, positive to negative remains unchanged, or none of the above, which might be the answer.
Option 2.
Option 2, positive to negative, right? So we have discussed that again. So this action potential in case of resting membrane potential, the gates are actually closed there. So and also when depolarization on the signal has to be transducted, then it starts to change its membrane potential, right? Depolarization happens when voltage gated sodium channel opens, where the sodium influx actually take place into the interior of the cell, and then it reaches a particular point. Afterwards, when it reaches a saturation point, it undergoes a process called as repolarization, where the sodium channels actually closes out and the potassium channels actually gets opened, and the potassium actually goes out of the cell, right? So this causes a dip in this positive potential range to a negative potential range, thereby restoring the resting membrane potential there. So the answer to this question is that during repolarization, membrane potential shifts from positive to negative value of in this particular step in the action potential there. So I think we have discussed this earlier also. Should I explain it once more, or is this clear, or anyone has any doubt? Okay, so I think it's quite clear. So I'll just move on to the next question. Coming on to question number 13. During depolarization, which ions move inside the cell from the outside? Sodium, potassium, both A and B, none of A and B. Depolarization.
Option 1.
Option 1, sodium, right? Yeah, we have discussed this earlier, because in case of sodium, in case of depolarization, case of resting membrane potential, voltage gated sodium channel as well as potassium channels are closed. In case of depolarization, voltage gated sodium channels opens out, right? So the sodium levels are more on the outside than compared to the inside of the cell. So when these voltage gated sodium channels actually gets opened, the sodium that is present on the outside of the cell or the environment, outside environment, moves into the cell, thereby causing depolarization there, right? That is the shift of membrane potential from the negative value to the positive value there. So during depolarization, which ions move? The sodium ions move from inside of moves inside the cell from the outside, that outside environment to the inside of the cell. So I think this is clear. And in case of depolarization, the opposite happen. Repolarization happen. The sodium channels close, the potassium channels opens out, and the potassium which is present on the inner surface of the inner content of the cell moves to the outside because of the opening of potassium channels there in case of repolarization. So I think this answer is quite clear. So does anyone have any doubt regarding this? Or should I explain the action potential again? Okay, so I think the answer is quite clear. So I will just move on to the next question. Coming on to question number 14. Which unit of muscle cannot be used for force measurements? Myotube, myofiber, myoblast, or none of the above, which may be the answer.
Myoblast.
Myoblast, right? Option number 3. Okay, good. So basically, myoblast is what you call as it's not the functional part, but it's a part of the cell, part of a muscle fiber that actually generates what is called as a myotube there, right? So satellite cell divides, proliferate to form myoblast. This myoblast arrange in a specific pattern and loses its identity to fuse together to form what is called as a myotube, which is a functional part of a myofiber, or what it forms what is called as a myofiber there. So basically, if you look into a myotube, it's the functional part. So if you have a myotube, basically you can what you can study a force measurements that are generated by contraction and relaxation of this myofiber, right? In case of myofiber, basically it's a collection of myotubes there. So basically it can also be used it can also contract as well as what you call as a relax, and therefore it can actually generate a force which can be used to study this kind of measurements, right? In case of a myoblast, it is actually the proliferating unit of a muscle fiber, right? So it cannot generate a force; it can just proliferate to form other myotubes and internal myofiber there, right? So it cannot generate any force there. So basically, the answer to this question, from that itself we can know that which unit of muscle cannot be used for muscle force measurement. Answer is myoblast. So anyone has any doubt? Okay, so I think it's clear. So coming on to question number 15. Neuromuscular junction is made at the junction of muscle and motor neuron, glia and motor neuron, hippocampal neuron and osteoblast, none of the above, which might be the answer. Sorry, there was a mistake here.
Option 1.
Option 1. I think everyone has the same answer. Option number 1. Muscle as well as motor neuron there. So the motor neuron are actually present within the peripheral nervous system, right? And the muscles that are associated with are sensory in nature. Motor neuron is basically used to carry out the sensory function and all. So basically, if you look into a motor neuron, when it joins to, there are two types of motor neuron, right? Alpha as well as gamma. Alpha is more probably associated with this, what is called as a extrafusal muscle fibers, right? While gamma neurons or motor neurons are associated with intrafusal muscle fibers there, right? So together these intrafusal as well as extrafusal muscle fiber actually helps the muscle forms what is called as a muscle spindle there, which actually has the muscle fiber to contract as well as relax there. And this particular gamma as well as alpha neurons that are connected to this particular muscle fibers actually when they are connected to this particular extrafusal or intrafusal muscle fiber, they form what is called as a intramuscular junction there, right? So when they form this particular intramuscular junction there, what happens is that several neurotransmitters are present there between this, sorry. Okay, several neurotransmitters are present there that actually sends the signals from this particular nerve ending to the muscle fibers, telling it to actually contract or relax there, right? So that's what neuromuscular junction is for. Basically, chemical signals actually transition between this particular junction that is formed there. Glial and motor neuron basically don't happen because glial cells are basically present within the central nervous system there, right? So that transition later take place. Actually take place within the peripheral nervous system and everything. Glial cells include, as I discussed, oligodendrocyte, astrocytes, as well as one more was there. Basically, I think 3 of them are there. 2 of them name I actually remember right now. And also hippocampal neurons actually present in central nervous system, and osteoblast actually bone forming cells, right? Osteoclasts are also there, osteoblasts are also there, and osteoblasts are involved in forming bones there. So the answer to this question is neuromuscular junction is made at the junction of muscles as well as motor neuron. So I think this answer is particularly clear. So does anyone have any doubt regarding this? Okay, so I think this is clear. So I will just move on to the next question. Coming on to question number 16. Bungarotoxin causes paralysis by blocking potassium channel acetylcholine receptors both A and B, and none of the above, which might be the answer. Okay, I'm getting option number 2, acetylcholine receptor. Any other option? Okay, so basically you're right. The answer to this question is acetylcholine receptor. So acetylcholine, as you know, is a neurotransmitter, right? So it is present in both central as well as peripheral nervous system, CNS or PNS in this case. And it has the ability to both stimulate as well as block a response. Not only to stimulate a response, it can also block a response there. So if you look into the function of this this particular acetylcholine, then it is involved in contraction of smooth muscles, it dilates blood vessels, it increases bodily function and slow heart rate basically. So basically, if you look into one of this particularly in nervating there, schematic diagram of nervating there, and what happening within a synapse there, this synaptic or neuromuscular junction there. So you can see that acetylcholine that is formed by different reaction like glycolysis and all. You can see that it then with the help of this choline acetyl transferase. This is also called as a chat enzyme there. Basically, we have discussed this particular answer before. With the help of this particular chat enzyme or choline acetyl transferase forms what is called as a acetylcholine there, and then it forms vesicles within this neurotransmitter, this neuromuscular region there, and then it exits out by fusing with the plasma membrane of the nerve ending there, and then it then interact this acetylcholine upon interaction with certain receptors that are present at the cell surface there, and then it activates or either carries out this particular functions involving upon binding it carries out the particular function involving contraction of smooth muscles, dilation of blood vessels, increasing bodily functions, loads, heart rate, etc. So basically, if you look into these acetylcholine receptors that are present on the cell surface, it consists of this particular subunits, alpha, beta, as well as gamma subunit there. And upon binding of this acetylcholine receptors, it actually opens up this particular channel that is present within this receptor there, and then it allows the movement of ions, influx of ions from the exterior of the cell to the interior of the cell. So that's what causing this particular what you call this particular function there, contraction, dilation, whatever is necessary, because it can either stimulate or block a response there, right? So Bankara toxin actually causes paralysis by affecting this particular function there. So whenever this particular function is affected there, this particular influx is not there, so this particular functions are getting affected there. Normal function that should be carried out within that particular muscle fiber or any other part of our body is not happening there. And so basically that's the basic function. It interacts with the acetylcholine receptors there, and thereby preventing the binding of this acetylcholine to this particular receptor, so the channel will is getting closed there, and then it does not perform this particular function there. So and it leads to paralysis in this case. So I think this particular answer is clear. So anyone has any doubt regarding this? And I think it's also irreversible in nature. I think it should be checked out. I also don't recall the particular answer to the question. So yeah, so I think this is quite clear. So I will just move on to the next question. Coming on to question number 17. For myotubes cultured on a cantilever device, to move cantilevers, which property of cantilever should be similar to that of myotube? Stiffness, color, weight, or width? Which might be the answer?
Option 1.
Option 1, right? Yeah, so basically you are right. So the answer to this question is option number 1, stiffness, right? So as I mentioned before, when you are developing a micro cantilever there, right, it should be flexible enough to move upon the contraction and relaxation of muscle fiber, and it should not be flexible so much so that it can break out within the afterwards for a long period of time, and it should also not be that much thick, because if it is thick, then oscillations might not, the muscle might be contracting, but the oscillations might not be present there, because the micro cantilever is not that flexible or sensitive to that oscillations there, right? So the thickness matters, the nature of the material that is being made, that is used to make that that particular micro cantilever there matters in this case. So the answer to this question is stiffness for myotubes cultured on cantilever devices. To move cantilevers, which property of cantilever should be similar to myotubes? The answer is stiffness, not the other option, color. Width is not there, because what matters is the thickness or the stiffness of that particular material. So I think this answer is clear. So anyone has any doubt regarding this? Okay, so I think this answer is quite clear. So I will just move on to the next question. Question number 18. Hippocampus is associated with movement, color vision, learning and memory, vocation. Which might be the answer?
Option 3.
Option 3, learning and memory. So I think everyone agree with that particular option, right? Learning and memory. This was also discussed earlier, also in the previous week. I think week before that, I think. So hippocampus is basically a part of central nervous system there, right? So it is essential for converting short-term memory into long-term memories. So and it stores these particular memories. So and it also helps to recall one's memory, right? If you have stored within that particular brain, then it should also have the capability to recall or revive or retrieve it back whenever the individual wants it to be there. Right? So hippocampus actually carries out this function. So it actually, if you are studying for a long time, if you are looking visually into something, then that gets incorporated into that particular memory, right? So that is converted into long-term memory, because it remains, if it remains a short memory, it can be lost for a, we can lose it for after some time, right? If it is converted into a long-term memory, then it can be stored and can be recalled back whenever it is necessary. So this particular function is carried out by hippocampus, that is by memory, and it also helps to carry out spatial navigation, helping individual to understand the environment and remember the location of the objective related to themselves. So that's also regarding the memory there, because for spatial navigation, because if you are waking up in the morning, you should know where other things are kept and everything. So that actually is carried out because of this memory storing capacity of hippocampus there. And also cognition memory should also be there from past experience and also other social interaction that you have done in your past life or recently. Then you need to learn something from that particular memory or experience that you are getting there, right? So that particular learning stage is carried out by this particular hippocampus also, and it also works with the particular region called as amygdala to link memories with emotion there. And this connection enhances the encoding of emotionally significant events, making them easier to recall later. As I said, recalling is one such function of this hippocampus, and this carried out particularly with this particular another region of the brain called as amygdala. So hippocampus is associated with not only in memory, storing or recalling memory, but also in learning from the past experience or any other social interactions that we earlier had. So that's the particular function of this hippocampus there. So anyone has any doubt regarding this? Okay, so I think this is quite clear. So I will just move on to the next question. Coming on to question number 19. What is a pungent pain-producing compound found in plants of the capsaicin family? It exerts excitatory, desensitizing, and toxic effects on subset of sensory neurons or motor neurons, including the polymodular nociceptor population. I am actually not sure what this means. So option number 1, capsaicin, TTX, digitoxin, as well as xanthin 14, which may be the answer.
Option 1.
Yeah, basically from the name itself, you know that capsaicin family actually consists of this particular, option number 1, particular compound present in that is pungent in nature, like chili and everything. So basically, it's not that like chili flavor like normal chili does, but it does provide this particularly pungent compound. And it is known for its ability to produce pain and other sensory effects, similar to what a chili produces there. And also, it primarily interacted with a subset of sensory neurons, particularly those expressing this particular receptor there, transient receptor potential vanilloid 1, TRPV1 receptor, which is actually classified as polymodal nociceptors there. So basically this effect is quite slow. It provides excitatory effect initially and leading to a slowly pain sensation there, followed by desensitization, as you have read from the question there, where these neurons become less responsive to further stimuli, including other painful stimuli and nauseous heat. So capsaicin is one such one such inhibitor that is similar to that of magaratoxin that you earlier discussed there, because it inhibits the activity of this particular receptor. It binds this particular receptor there and then inhibits somehow its activity there, thereby leading to desensitization there, and and also causing less response to particular stimuli there. And I hope this answer is clear. Capsaicin is a pungent poem-producing compound found in plants of the capsicum family, exert excitatory, desensitizing and toxic effects on a subset of sensory neurons. These particular neurons, that is receptors that are found in this neuron, that is transient receptor potential amyloid 1 receptor or TRPV1 receptor, including this particular receptor population there. So I think this answer is clear. So does anyone have any doubt there? Okay, so I think this answer is quite clear. So coming on to a final question, that is question number 20. What of these compounds, that is TTX, 4AP, calcium or free radical, blocks calcium channels, sodium channels, sorry, which might be the answer? Okay, so I got option number 1. So you are basically like tetrodotoxin, right? So tetrodotoxin is a compound that blocks sodium channels. So sodium channels is what is essential for causing depolarization there, right? And it inhibits this voltage gated sodium channel in a highly potent or specific manner. It is highly active in nature and preventing the sodium ion inflow, thereby preventing depolarization there. And also if you look into the other option, 4AP or 4-aminopyridine blocks potassium channels, not sodium channels. So if potassium channels is blocked there by in the presence of 4-aminopyridine, what happens is that repolarization don't happen, right? Potassium channel won't move out into the extracellular environment of the cell, right? And yeah, so other components like calcium and free radicals are not probably involved there. Free radicals are basically providing like oxidative stress there, right? So in case of cell culture, free radical actually degrades out this cell, so chances are that we don't want that in this case. So yeah, so that's the basic function there. For tetrodotoxin blocks sodium channel, while additionally 4AP, 4-aminopyridine blocks potassium channel. So I think that answer is quite clear. So anyone has any doubt regarding this? Okay, so I think this is quite clear. And so I think this is the last question. So thank you for everyone for joining. So does anyone has any final doubts regarding this question, regarding the whole section that we have just took today basically, sorry. Okay, so I think that's quite clear. So thank you for joining and have a nice day, everyone. Yeah, thank you. And good luck for your exams also.
Thank you, sir.
Thank you, sir.